On the 17th of May, 2024, an event took place on the San Antonio River that would completely transform the area. Early that morning, a group of biologists drove to a site near the outlet of a flood control tunnel. The atmosphere was tense, and every eye was fixed on the small containers being carefully lowered to the ground. Inside were dark brown, motionless objects. They were freshwater mussels. "This is history," one expert whispered to the camera. It was the first time they had been released back into the wild in Texas. "You may only know them by name, but one day we may have to thank them for everything they do." Biologist Austin Davis put on a pair of gloves, picked up each mussel, measured the length of its shell, and recorded the information. Some individuals were even fitted with something tiny and blue. After that, employees from the San Antonio River Authority waded into the water, carried trays of mussels, and carefully placed them by hand into gaps between the rocks on the riverbed. By the end of the year, more than 3,500 mussels had been released. But what exactly was their role? And that was not the only mystery. Why did an ecological restoration project that had been widely promoted during its public announcement later keep the release location secret? If you live in the United States, you probably already know how important the mighty Mississippi River is. But today, I want you to focus on another special location, Mission Reach. For decades, in the name of flood control and urban development, people transformed this section of the river into a dry, concrete channel designed to move water away as quickly as possible after the devastating floods of 1921 and 1946. What was the result? The natural riverbanks disappeared, while trees, gravel, rocks, and shelter for fish and other aquatic animals were wiped out. Although Mission Reach has recently been brought back to life with lush green vegetation, did you know that beneath the water, which appears healthy on the surface, the ecosystem is still largely empty? Freshwater mussels have disappeared from the riverbed. Without them, the river is still like a body missing its lungs. But we will explain their specific role later. The problem is that freshwater mussel populations did not disappear because of just one cause, but three. The first was the fashion boom of the late 19th century, when mussel shells were harvested on a massive scale to make buttons. In 1899 alone, approximately 16 million pounds of mussel shells were collected in Wisconsin, wiping out mussel beds that had taken centuries to form. Next, hydroelectric dams transformed free-flowing rivers into still reservoirs, reducing oxygen levels beneath the sand and gravel while cutting off the migration routes of fish. Finally came the devastating invasion of zebra mussels from Eastern Europe in 1988. They attached themselves in dense clusters to native mussels, preventing them from feeding, breathing, or reproducing. As a result, this species, which once lived along approximately 850 kilometers of the Mississippi River and 9 of its tributaries, lost more than half of its historical range and was federally listed as endangered in 2009. Even after numerous conservation measures and harvesting bans were introduced, its population showed little sign of recovery. At Mission Reach, 21 locations were surveyed between 2015 and 2017, but only one living individual was found. If no one was harvesting or harming them anymore, why couldn't they simply return on their own? The truth is that adult mussels can move only extremely short distances. They cannot crawl for miles across fragmented sections of river, while the remaining original populations are already far too small. But the most important reason lies in their highly unusual life cycle. The larvae produced by female mussels cannot simply grow on their own on the riverbed. They must attach themselves as parasites to the gills or fins of the correct host fish species. Only there can they develop, transform, and eventually drop to the bottom as juvenile mussels. When those fish species disappeared because dams blocked their migration routes, the entire reproductive cycle was completely broken. Clearly, the structure of the ecosystem had changed so much that nature could no longer heal itself. But the story of Mission Reach continued. In 2017, the U.S. Geological Survey partnered with the San Antonio River Authority to conduct a study at Mission Reach and determine whether the river was truly ready for freshwater mussels to return. Mussels were placed at 2 sites within Mission Reach and at 1 control site in Goliad County. The 4 species being monitored were yellow sandshell, threerage, pistol grip, and pimpleback. To everyone's surprise, the mussels recovered in October 2020 brought an extremely encouraging sign. The official report, published in 2021, recorded survival rates of 75% at the first mission reach site and 81% at the second, for an average of about 78%. Meanwhile, the downstream control site achieved a survival rate of only 50%. Mussels at one mission reach site also showed noticeably stronger growth. This indicated that the water quality and riverbed conditions in this urban section of the river were sufficient to support adult mussels. The researchers did not stop at proving that adult mussels could survive. They continued by keeping juvenile mussels in San Antonio River water for 28 days, followed by 48 hours of exposure to stormwater runoff. The preliminary test recorded a 100% survival rate, although potential risks from copper, ammonia, and urban runoff still needed to be monitored. Although these findings could not guarantee that the reintroduction would succeed, they left the research team extremely encouraged. After years of restoring mission reach, freshwater mussels could finally survive there again. But to achieve this rare, positive sign, people had to begin restoring the river many years earlier, and that is the next part of the story. Before placing a single mussel on the riverbed, they first had to rebuild the physical structure of the river itself. The Mission Reach project was completed in 2013 as part of the $384.1 million San Antonio River Improvements Project. It extended for 13 miles, with approximately 9 miles receiving habitat improvements. Engineers recreated riverbends, sequences of deep pools, steady-flowing runs, and shallow riffles. They also reconnected 2 historic river channels and restored areas of slow-moving water along the river's edge. Along both banks, more than 20,000 native trees and shrubs were planted across nearly 334 acres, an area larger than 250 American football fields. Their roots stabilized the soil, their canopies provided shade, and the restored variations in water depth and current speed brought back shelter for fish, insects, and other bottom-dwelling organisms. From 2013 to 2016, approximately 84,000 Guadalupe bass were released into Mission Reach and the San Juan remnant. Later genetic testing showed that they had successfully reproduced, an encouraging sign that the river was beginning to sustain life on its own. Fish, birds, and plants gradually returned, but the riverbed was still largely empty. Management officials acknowledged that freshwater mussels were the critical missing piece needed to complete the restoration. From 2014 through the fall of 2019, biologists surveyed more than 226 miles of river and inspected over 200 locations. They documented and released more than 1,000 mussels back into the water. The researchers identified each species, collected small tissue samples to study genetic lineages, and searched for parent populations with enough genetic diversity to support captive propagation. At the same time, the researchers realized that these efforts alone were still not enough. They wanted freshwater mussel populations to recover more quickly. Another reasonable theory was that if the right fish species were present, the environment would also be better suited for mussels. So the research team also studied gar, catfish, bass, sunfish, and shiners that could serve as hosts for mussel larvae. Spotted gar were used to propagate yellow sandshell mussels, while flathead catfish were included in several other trials. Surveys at Mission Reach recorded 5,278 individual fish, representing 28 species, 88% of which were native. In a single netting pass covering approximately 20 meters, the team captured 13 long-nosed gar and spotted gar, an important sign that the fish needed to support the mussel's life cycle were present. Once they confirmed that the conditions were stable, the 2024 freshwater mussel release you saw at the beginning finally took place. Since we have already covered that event, we will not repeat it here. Now we will get to the question that both you and I are most curious about. What purpose can creatures that look like rocks possibly serve? You know, oysters, right? Freshwater mussels work in a similar way, except they live in freshwater lakes and rivers rather than in the ocean. Mussels draw water through their gills to feed, while also trapping algae, phytoplankton, bacteria, fine sediment, and suspended organic particles. The U.S. Fish and Wildlife Service estimates that a single mussel can filter about 5 to 10 gallons of water per day. Under favorable conditions, some species may filter nearly 15 gallons, or about 57 liters. Using the conservative estimate of 5 to 10 gallons for the 3,500 mussels that were released, the population could theoretically filter about 17,500 to 35,000 gallons of water per day, equivalent to roughly 66,000 to 132,000 liters. At the maximum rate of 15 gallons, that figure could approach 199,000 liters. However, this is only an illustrative calculation, not a result that has been directly measured at mission reach. The actual filtration rate depends on the species, size, water temperature, amount of algae, current speed, and number of mussels that survive. More importantly, mussels do more than simply remove material from the water column. They digest part of it, store nutrients in their bodies and shells, and transfer the rest to the riverbed as biological waste. This makes energy that was once drifting through the water more accessible to bacteria, aquatic insects, and other bottom-dwelling organisms. Studies describe this process as an important link in a river's nutrient cycle and food web. When present in high enough numbers, their bodies buried in sand and gravel can also help hold sediment in place and make the riverbed more stable. The shells of living mussels, as well as the shells left behind after they die, create hard surfaces, small crevices, and shelter for insects, snails, and many other invertebrates. Mussels themselves also become food for fish, birds, and mammals living along the river, but do not mistake them for a water treatment plant. Mussels cannot turn toxic wastewater into clean water, remove every chemical, or save a river that has lost its oxygen. They themselves need sufficiently clean water, a stable riverbed, and the presence of suitable host fish in order to survive. That is why the greatest value of these mussels is not simply the amount of water they can filter. If they survive, grow, and reproduce over many years, it will provide clear evidence that Mission Reach is not only becoming greener on the surface, but that the ecosystem beneath the water is truly recovering as well. One more thing. The project's original goal was to release as many as 5,000 mussels, but by the end of the year, only 3,500 had been released. That gap reflects just how demanding the breeding process is and how difficult it is to produce a reliable supply. Most of the mussels were raised at the Inks Dam National Fish Hatchery in Burnet, Texas. The mussel propagation program there began developing in 2017. By 2019, the U.S. Fish and Wildlife Service and the San Antonio River Authority had established a dedicated partnership for Mission Reach. The San Marcos Aquatic Resources Center studied propagation techniques. The Southwestern Native Aquatic Resources and Recovery Center conducted genetic analyses, and Inks Dam handled large-scale production. Everything begins with a kind of biological performance. And yes, that description is not an exaggeration. To attract a host fish, a female mussel extends part of her soft tissue outside the shell, perfectly imitating the movements of a small, injured fish struggling in the water. When predatory fish, such as bass, rush in to attack the decoy, the female immediately releases hundreds of thousands of Glochidia larvae directly into the fish's mouth and gills. Under the scientists' microscopes, these tiny larvae clamp tightly onto the fish's gill tissue. The surrounding tissue then grows around them, forming something like a protective pouch. Over the next several weeks, they live as parasites, drawing nutrients from the host fish while developing a foot, a shell, and the organs needed for filter feeding. Once the transformation is complete, the juvenile mussels detach from the fish's gills and fall to the bottom of the tank. This is a life or death stage with an extremely high mortality rate. At this point, juvenile yellow sandshell mussels measure only slightly more than 1 millimeter in length. To prepare them for life in the wild, specialists must raise them until they are about 20 times larger, reaching roughly 20 millimeters before they are ready for release. Before being set free, they are placed in floating nurseries, sand-filled baskets, positioned directly in the San Antonio River. River water flows through the baskets, allowing the young mussels to adjust to natural temperatures, microorganisms, and algae, while remaining safely protected from predators. Choosing the right time for release is extremely difficult. If they are released too early, strong currents may wash them away. If they are kept in the laboratory for too long, they may lose their ability to adapt to natural conditions. These year-to-year fluctuations in breeding success explain why the original goal of releasing 5,000 mussels has not yet been fully achieved. But there was another issue. Do you remember the question we asked at the beginning? Why would a major ecological restoration project receive extensive media coverage only for officials to keep the actual release locations completely secret afterward? The truth is that the entire press event, filmed on the 17th of May 2024, was held at a demonstration site. The real locations where more than 3,500 mussels were released were removed from every publicly available map. Can you guess why? Scientists at the San Antonio River Authority had to take this step to protect these young environmental engineers from human curiosity. If the locations were leaked, crowds of curious visitors could arrive and accidentally trample the riverbed, disturb the sediment, or even pick up the mussels for a closer look. Any of these actions could kill these highly sensitive animals. This secrecy created an unusual situation. The public knew that freshwater mussels had returned, but their actual habitat beneath the San Antonio River remained a mystery. However, even though their locations were hidden from people, scientists still had a way to observe their lives deep beneath the water using a special technology. About 25% of the mussel population, at least 875 individuals, were fitted with tiny PIT tags that function like electronic passports. The remaining mussels without chips were manually marked on their shells for identification. Each chip contains a completely unique identification code. With this network of electronic passports, specialists can use dedicated scanning equipment to track individual residents beneath the river. Are they still alive? How many millimeters have their shells grown? Did the latest flood carry them far from the release site? This careful monitoring creates an invaluable scientific database, helping biologists evaluate the success of the project and prepare for future releases. But what about the other 75%? How can scientists monitor mussels that do not have electronic tags? The answer is simple. When technology is unavailable, they rely on painstaking manual methods. To avoid overlooking any individuals, specialists from the U.S. Fish and Wildlife Service must conduct extremely detailed underwater surveys. They dive to the riverbed with PIT scanners, search through individual sections of sediment, and follow strictly timed survey procedures. Their goal is to relocate mussels identified only by the markings carved into their shells so they can measure growth and calculate survival rates. Although the project currently remains limited to the release of 3,500 mussels, with no clear sign that additional releases will take place, and although the mussels appear to be developing steadily, the project still cannot declare victory. Biologists from the U.S. Fish and Wildlife Service and the U.S. Geological Survey remain on high alert because, behind the veil of mystery beneath the San Antonio River, 5 ticking time bombs are still waiting to threaten the survival of the reintroduced mussels. The most immediate danger is the harsh nature of an urban river. Water pouring off streets and parking lots into Mission Reach can cause sudden surges in flow, sweeping mussels away from their release sites, burying them beneath mud, or destroying the sand and gravel substrate they need to survive. The threat of pollution is also very real. The River Authority previously recorded copper concentrations in stormwater runoff that exceeded the EPA's acute water quality criteria at the beginning of a 2016 rain event, while also identifying copper and ammonia as 2 substances requiring particularly close monitoring. At the opposite extreme, prolonged drought can drastically lower water levels, raise river temperatures, and reduce dissolved oxygen. In-depth studies from the U.S. Geological Survey warn that increasing heat can drain the energy mussels need for growth and reproduction. Severe droughts in the past have even wiped out mussel populations or completely reshaped their distribution across many streams. In addition, if essential host fish such as Guadalupe bass disappear from the area, the mussel's life cycle will immediately break down because their larvae will have nowhere to attach and develop. Then there is the continuing threat of urban pollution, as harmful substances such as oil, grease, and heavy metals are washed from city streets into the river by stormwater, quietly contaminating the water in ways that are difficult to control. Just as dangerous is the genetic challenge. If the breeding population comes from too few parent mussels, a lack of genetic diversity could leave future generations extremely weak, making them more vulnerable to disease and less capable of adapting to extreme changes in weather. Because of these surrounding risks, the success of the restoration cannot be measured through short-term reports. Instead, it must pass a demanding 5-level evaluation of long-term survival. At levels 1 and 2, the mussels must survive through multiple harsh seasons, while their shells must continue growing steadily, millimeter by millimeter, proving that their bodies are successfully absorbing food from the natural environment. At level 3, they must demonstrate that they can remain firmly established and bury themselves securely in the riverbed without being swept away by powerful currents after major storms. Finally, the project's ultimate victory lies in levels 4 and 5. The mussels must naturally begin attracting host fish, complete the transformation stage, and produce a new generation of juvenile mussels that fall to the riverbed and grow into a stable, self-sustaining population, without requiring constant restocking from the Inks Dam National Fish Hatchery. The effort to revive the San Antonio River clearly remains a long and difficult journey. Humans are still watching every breath of the river, hoping to prove that when nature is finally given back what it needs, it can find a way to heal itself. In the end, the San Antonio project shows that restoring nature is never as simple as releasing animals into the water and waiting for a miracle to happen. Before freshwater mussels could return, people had to reshape the river, replant vegetation along its banks, improve water quality, restore the sand and gravel riverbed, and bring back the fish community. More importantly, they had to rebuild the invisible relationships between species relationships that had been broken for decades. This story also reminds us that a greener river is not necessarily a complete ecosystem. Flood control structures once protected people, but at the same time, they also destroyed a great deal of natural habitat. And sometimes, the creatures that receive the least attention are the ones that play the most important roles. Think back to the scene of the biologist bending down and placing each mussel carefully onto the riverbed. The action may have looked small, but behind it were hundreds of miles of surveys, years of captive propagation, and an entire decade of restoration work at Mission Reach. Do you think this project will succeed? Leave your thoughts in the comments and subscribe to the channel to follow more real stories of nature restoration.